Hello, my name is Charles Wilmot. I am currently a PhD student at the Goethe University of Frankfurt, and I work at the Frankfurt Institute for Advanced Studies. Today I will be presenting our paper called Self-Calibrating Active Binocular Vision via Active Encoding with Deep Autoencoders. In this talk, we present a model of the self-calibration of active binocular vision that formulates the task as an intrinsically motivated reinforcement learning problem. In contrast to classic computer vision solutions to Vergence control and object tracking, our model does not require predefined visual representations or kinematic models. Instead, it learns from scratch from raw images. Nevertheless, it achieves sub-pixel accuracy in its Vergence and tracking movements, demonstrating successful self-calibration. Already a few minutes after birth, newborns can open their eyes. However, it takes about 3 months for infants to learn to perform Vergence accurately. In our paper, we try to answer the questions: how does the brain learn active vision, and how can we model this learning process in a computer? The hope is that by better understanding the way active vision develops, we might also gain insight about the acquisition of other skills. Human vision is an active process and comprises a number of different types of eye movements. The first type of eye movement is called version. It corresponds to all conjugate movements of both eyes. The joint horizontal movement toward the right is called the dextroversion, and towards the left the levoversion. Joint vertical movements are called supraversion and infraversion. The eyes also have the ability to rotate around the axis of vision. These movements are called dextrocycloversion and levocyclovergence. The second type of eye movement is called vergence and denotes equal and opposite movements of both eyes. It comprises the convergence and divergence movements, rotating the eyes inward and outwards, and the encyclovergence and excyclovergence, rotating the eyes in opposite directions around the axis of vision. Version and vergence movements enable to focus the gaze on objects, pursue them as they are moving, compensate for the movement of the head and the body within the scene, and to perform saccadic eye movements to direct the attention. How the human visual system calibrates itself, learns to make use of the version and vergence movements, and learns the required sensory representations of the visual input signals is only poorly understood. A better understanding of this process might allow to build fully self-calibrating active vision systems that are robust to perturbations, which could in turn form the basis for models describing the autonomous learning of object manipulation skills such as reaching or grasping. Learning active vision is relevant in the context of developmental robotics because vision is a cornerstone of infant development. It conditions motor skills learning, as visual information is useful to perform object-directed movements like grasping and reaching. Binocular vision also causes stereopsis, which is relevant for learning to semantically segment objects and for learning abstract representations of the scene. Stereopsis refers to the perception of depth and three-dimensional structure obtained on the basis of visual information deriving from binocular vision. On that schema, the point P is being focused by both eyes. Its image is projected onto the foveas. The point Q is projected on different points on the retinas, but their eccentricity with respect to the foveas are the same. The brain interprets that the point Q is at the same depth than P. The images of R, however, do not have the same eccentricity on the retina. It results a vergence disparity between the two eyes, and the brain deduces that the point R is closer than P. Let us have a quick look at this schema of the human visual pathway. You probably recognize here the two eyes and their optic nerves, the optic chiasma, the lateral geniculate nuclei, and the visual cortex. I want to draw your attention on the fact that similar information projects on the same brain region. In particular, the information about the left part of the visual scene from both eyes is grouped at the optic chiasma and then projects on the right LGN, and vice versa for the right part of the visual scene from both eyes, projecting on the left LGN. At the LGN, we can differentiate two pathways of information. The first is the so-called parvocellular pathway. It conveys spatial information like colors at a high resolution and is sensitive to even low contrasts. The second pathway is the magnocellular pathway, which carries information with a much lower spatial resolution. It is sensitive to high contrast only, and it holds information about motion. I would like now to introduce the principle of efficient coding proposed by the neuroscientist Horace Barlow in 1961. It postulates that in order to reduce high energy demands from the brain, neurons tend to reduce the number of spikes needed to transmit information. A good way of doing so is to encode data and to transmit the compressed representation. The idea of efficient coding is thus that neurons convert the information into a less informative and more abstract code to save energy. An extension of this principle, called active efficient coding, has been formulated, proposing that the behavior itself can produce sensory information easier to encode. When applied to the visual system, active efficient coding predicts that the eye muscles must be controlled by the brain in order to produce redundant, and thus easily encodable, visual information. Increasing the redundancy between the two eyes leads to focusing on the same point in the environment, and increasing the redundancy in time to tracking of objects. As a consequence, learning both vergence and tracking boils down to a redundancy maximization task. But what is redundancy exactly? So, the redundancy between two random variables can be defined as the mutual information between them. This quantity is, however, often intractable. The solution proposed by active efficient coding algorithms in order to alleviate this problem is to measure the redundancy via the reconstruction error of a lossy compression algorithm. Previous implementations of active efficient coding used a sparse compression algorithm called matching pursuit. In this research, we proposed to replace it with autoencoders. Additionally, we proposed an improvement of the reinforcement learning reward signal. Let us now focus only on the case of Vergence learning. The autoencoder jointly encodes the left and right images from a robotic camera. Our hypothesis is that perfectly aligned left-right pairs have a lower reconstruction errors than pairs with a Vergence disparity. The encoded visual information is then fed into a reinforcement learning algorithm. We used a value-based algorithm for its simplicity, namely the deep Q network algorithm. The reward for training the Q network is the difference between the reconstruction error before and after performing the motion, meaning that we reinforce actions bringing the agents from a high reconstruction error to a lower reconstruction error. It is an improvement over the previously proposed rewards, consisting of only the negative reconstruction error after performing the motion. Both the newly proposed reward and the older one are in many ways similar to intrinsic motivations proposed by Jürgen Schmidhuber as a model of curiosity in the early 90s. In these researches, the agent learns a model of its environment as it explores the world, and the reward of the reinforcement learning algorithm is defined as the error of the prediction model. So the agent is learning to experience poorly predicted state transitions. In our approach, the model does not learn to predict the future, but simply encodes the sensory information instead. I now would like to detail a little bit the design of the encoding mechanism. We used a very simple convolutional autoencoder, whose filter size is 8 by 8 in the first layer and 1 by 1 in the following layers. Effectively, this is equivalent to encoding 8 by 8 binocular patches extracted from the camera data using a fully connected autoencoder. We also imitated the way the visual acuity in human vision decreases as we move away from the fovea. We did so by encoding two 32 by 32 pixels region extracted at the center of the camera images. The first is called the fine scale and it covers an angle of 9 degrees, corresponding to high acuity. The second is the coarse scale and it covers an angle of 27 degrees, corresponding to a lower acuity. To justify this design choice, I prepared a little animation showing an 8 by 8 pixels binocular patch taken from the fine scale. As we rotate the left eye only, the pattern on the left retina is shifted until the two images are completely different. When the Vergence disparity gets too big, the information about the direction in which the eyes must move disappears. It is also predictable that for such disparities, the reconstruction error of the patches does not reflect the error of the joint anymore. The use of a coarser scale alleviates the problem, while also being more similar to biological eyes. A movement of 3 pixels at the fine scale corresponds to a shift by only 1 pixel at the coarse scale. So far, I presented the parvocellular pathway of our approach. The magnocellular pathway, learning object tracking, is very similar, the only difference being that it encodes 2 consecutive binocular pairs instead of only 1. The learned abstract code then serves as an input to 2 deep Q networks, one for the tilt joint controlling vertical movement of the eyes and one for the pan joint controlling horizontal movement. I will now detail the experimental setup and present the result of this approach. You see here the simulation software we used, in which we placed a robot head consisting of 2 camera eyes. The head is placed in front of a screen which can move in all directions. In each episode, the position, speed, and direction of movement of the screen are sampled randomly. the tilt and pan joint of the eyes are reset to 0 degrees and the Vergence angle is sampled randomly. Each episode consists of 10 iterations of movement. The view angle of the camera and its resolution determine the angular size of 1 pixel, which is in our case equal to 0.28 degrees. In the following, we will measure all angles and angular speeds in pixels rather than in degrees, as it makes the results more interpretable. I will now present a few figures showing the performance of the algorithm. The first figure that I want to show is used to check the hypothesis that errors in Vergence or tracking result in an increase of the encoding quality. For each joint, we positioned the agent in order to control precisely the joint's error and recorded the reconstruction error of the autoencoder as the joint error varies. This plot thus shows from left to right the reconstruction error as a function of the pan, tilt, and Vergence error. The individual blue lines correspond to different images projected onto the screen, and the red curve shows the mean for all images. As you can see, the results show that the reconstruction error is indeed minimal when the joint error is minimal, thus confirming the hypothesis. The second figure details the composition of the reconstruction error, showing that of the fine and coarse scale. As you can see, for big joint errors, the reconstruction error of the fine scale stops increasing while the coarse scale is still affected. Next, I present the quality of the learned policy. In the same controlled setup as earlier, we recorded the preferred action as a function of the joint error. The red curves show the ideal actions. The results show that the agent consistently selects the optimal action that will decrease the joint error. This figure shows the speed at which the agent recovers from an initial joint error. What you see here is, for each joint, the mean absolute joint error recorded during 20 iterations of performing the agent's behavior. The results are averaged over many initial errors, ranging in -4 to 4 pixels. The dashed horizontal line represents the 1 pixel limit. As you can see, in average, the agent needs no more than 2 iterations to recover the initial error with a sub-pixel accuracy. Finally, this figure shows the performance of each joint as a function of the algorithm's training time. Again, the horizontal dashed gray line indicates an error of 1 pixel. It takes about 10,000 episodes to reach that level of performance. I will now present videos showcasing the performance of the approach. Both show a blue-red anaglyph of the left-right eyes of the robot. This helps visualizing the regions of the scene correctly fixated by the agent. The first video will show the quality of the learned Vergence policy. You will see a screen placed 2 meters away from the robot's head. We then simulate the agent's behavior, starting from different initial Vergence errors. The initial Vergence error written on the video is expressed in pixels. The two central squares represent the fine and coarse scale, each being sampled at 32 by 32 pixels. As you can see, the agent successfully recovers from even big initial Vergence errors. The same process can be repeated for testing the tilt and pan policy. In that case, the initial error indicated on the video is expressed in pixel per iteration. During the training procedure, the maximum speed of the screen is 4 pixels per iteration. The robot is, however, capable of successfully tracking faster objects, as shown in the video. The next video shows the environmental setup used during training. The agent's policy is performed for 30 iterations before the environment is reset. As you can see, the agent quickly fixates the screen in the field of view and tracks it as it is moving. I now would like to conclude. So we applied an extension of the efficient coding principle, the active efficient coding, to the learning of active vision. The resulting approach learns jointly a representation of the visual information and accurate eyes movements, successfully fixating objects and pursuing them as they move. In the future, we would like to combine the learning of active vision with motor control. We believe that this will enable the learning of visuomotor contingencies as the agent learns to track its own body parts. Another interesting direction for future work is to consider other sensory modalities. Unpublished work is revealing that active efficient coding can also be used to model the self-calibration of echolocation in bats in the auditory modality. More generally, the combination of different sensory modalities is an interesting topic for future research. Arguably, a key step in cognitive development is discovering and understanding the relationship between sensory signals from different modalities in order to arrive at a unified representation of the world. This work has been supported by the European Commission under the frame of the Horizon 2020 initiative.